Well, thank you, Ray. It's great to be here and to be able to participate in this really important meeting. This is going to be primarily a cancer talk rather than an antibody talk, but what I would try to do as I go through this is to identify some of the opportunities for antibody engineering that I think are currently limiting the field of spatial systems biology. And so I hope that one of the things I accomplish at this talk is to inspire some of you to help solve some of the important reagent problems that we're currently dealing with. Okay, I'm obligated to show you that I'm conflicted at several levels. Let you read that for a moment. And then move on to the general theme of this presentation. And that is that one of the things that we are attempting to do in cancer today is to think about cancer not necessarily as a disease of a gene, but rather as a heterogeneous adaptive system. And one of the things that you will appreciate is that when you think about cancer, you think about the cancer cells themselves and the fact that the regulatory circuits that comprise those cells and that drive their biology, their proliferation, their apoptosis, and so on, are typically deranged by genomic abnormalities. So we tend to spend a lot of time focusing on that. But in fact, the way that this cancerous system operates is through a remarkably complex exchange of signals between the environments in which the cancer cells sit and the diverse environments to which they move as they disseminate throughout the body. And it's actually the integration of these intrinsic and extrinsic environments that really determine how cancers are going to progress and how they are going to respond to therapy. And so we need to understand the whole system if we're going to come up with robust strategies to manage the system. So what I'm going to do in this talk today is to divide it really into two major pieces. I thought I would start with just a little bit about cancer genomics, since that is really, I think, what has gotten us to the understanding that we are today. Talk about cancer as a genomic disease, and then spend the bulk of the time talking about cancer as this heterogeneous adaptive system that I just described. And so as I go through that, I'll make the point that I think if we're really going to understand those kinds of systems, we're going to have to make heavy use of multiscale imaging to really be able to understand those interactions. I'm going to give you a couple of vignettes about how it is that information about the spatial organization of cancers have led us to new strategies to improve cancer treatment, both by managing intrinsic heterogeneity and by managing the extrinsic heterogeneity. So let me start with cancer as a genomic disease. And I think that you're all well aware of the fact now that for the last 5 years or so, there has been a major international effort in trying to define the omic landscapes of cancers. And this effort has basically come to its first, sort of the end of the beginning. I think that we do have good information on most of the major cancer types at this point. Maybe 25 or so thousand tumors have been analyzed for changes in genome copy number, mutations, structural changes, et cetera. And so we understand, I think, a lot of what comprise the working components or the deregulating components of cancers. And I thought I would just give you one vignette to try to set the stage for what follows. And to do that, just to summarize a little bit about what we've learned about breast cancer. And it turns out that the cancers in general, and breast cancer in particular, turn out to be remarkably complex. Breast cancers can be comprised of about 45 different regions of recurrent genomic abnormality. If you divide them into subtypes, you can define at least 10 subtypes that grows more or less regularly. One of the things that's pretty remarkable is that the somatic mutation rate in breast cancer is about 1 mutation per megabase. Now that's interesting in the sense that that means that a typical cancer will have a few thousand somatic mutations, some of which are targets for vaccines. And if you, the last time I heard Mike Stratton from the Sanger Center summarize mutational discovery in human cancers, I think we're up to 8 million different somatic mutations that have been discovered in human cancers at this point. So there is a lot to learn about here. Now, the general rule here is that in any particular cancer type, a few of the mutations are relatively recurrent, and most of them are rare. And in fact, most of these mutations that we've discovered have been discovered only in one cancer. So it's quite a diverse repertoire of things that we have to deal with. We also know that new mutations arise and are selected for as cancers progress under treatment. But I think that there is now a lot of functional evidence emerging from the field that a goodly number of these mutations are actually functionally important, maybe as many as a quarter. So this means that there is this vast space of unknown biology out there that we're still having to come to grips with and to understand how all of these mutations interact together to try to develop the phenotypes that we're trying to manage clinically. Well, it's obvious that reagents to study these mutations are, to first approximation, completely missing, but a real opportunity for antibody development as we go forward and as we begin to understand which ones are the real strong drivers in cancer. But all of that said, I think that okay, guys, you're going to have to, all right. One of the things that has come out of this whole business is a stimulation of the development of therapeutic agents, antibodies and otherwise, to attack some of the driver aberrations. And the thing that motivates the rest of this talk is that responses to these aberrations are certainly variable between patients and especially in advanced disease are not durable. And so one of the things that we have to do is to move from this sort of parts list to the assembly manual to, I think, understand how to develop more durable therapies. Next slide, please. Okay, so that takes me to then trying to understand cancer as this heterogeneous adaptive system rather than just a collection of broken parts. Next slide, please. All right, in order to do this, is there any chance you can actually fix this? Okay, so thinking about cancer as a heterogeneous adaptive system, one of the things that you will appreciate is that heterogeneity actually exists at many scales, sort of from the tissue level from the tissue level all the way down to the level of molecular structure. And one of the things that is really remarkable in, I think, science today are the new imaging modalities that are now emerging that pretty much allow us to study with microscopy features that range all across this almost 9 order of magnitude dynamic range from angstroms to centimeters. And so one of the things that we've been doing up in Portland recently is to try to put together the suite of instruments that will allow us to span this entire size range. And so, this starts at the high end with electron microscopy, moves up to super resolution light microscopy, and then anatomic imaging. And one of the points that I'll keep coming back to throughout all of this is that the technologies are present. What's actually not so well developed are the analytical workflows, and in particular the affinity reagents that you need to be able to inform yourself about what it is that you're looking at using these methodologies. But that said, the technologies themselves are there, and the computational strategies are emerging to allow us to manage these multiscale images that come out of this. And so we have as I said, assembled all of these into a multidisciplinary research building recently completed in Portland that brings together people with expertise in chemistry, antibody technology, imaging, computational biology, and so on, to try to bring to bear all of these analytics to cancer understanding and to cancer management. And so I'm going to tell you a little bit about some of the things that we've learned. And I'm going to start with this issue of managing tumor intrinsic heterogeneity. And you really don't get an appreciation of the extent to which this exists until you begin to apply antibody-informed diagnostics, imaging, if you will. And so what I'm showing you in this slide is on the left a image of a normal breast tissue stained for antibodies that inform on the differentiation state of the cells, the luminal cells, the basal cells, the fibroblasts, and so on that make up the normal tissue. As you can see, is well organized. When you get into cancer, of course, you lose the disorganization, and so on the right you'll see a breast cancer that would normally be classified as estrogen receptor positive. And you can see the fibroblast staining red and the tumor staining for keratin 19, a rather homogeneous tumor in this case. This tumor would be classified the same way, but in fact what you see now is the emergence of differentiation state heterogeneity. This would still be classified as a estrogen receptive positive tumor, but it has this basal-like component in it. If you go to what we call triple negative or estrogen receptor negative, HER2 negative breast cancer, what you see is incredible heterogeneity in differentiation status. Now, the question is that heterogeneity functionally important? And so one of the things that we've been trying to do of late is to model some of the heterogeneity, in this case using breast cancer cell lines. And one of the things that we've come to appreciate is that even cancer cells grown in the laboratory on plastic or in other different environments also display this same kind of differentiation state heterogeneity. They also model many aspects of cancer genomic aberrations. They model aspects of cancer pathway usage and so on. But the nice thing about these model systems is that they can be readily manipulated to ask the question, do those different subpopulations in the cancer respond to treatment differently? Now, I will say that we're using a 3-point definition of heterogeneity at this point. We're fully limited at this point by the availability of informative antibodies that could make this a more robust assay. But what we already know is that this is an informative approach. The heterogeneity persists in essentially all models, organoid cultures, xenografts, and so on. So it's a robust phenotype, and so we wanted to ask the question, does it have an impact on cancer response? And so in order to get to that, since we're easily able to manipulate these cells in culture and to treat them with various drugs, we ask the question, does the differentiation state change as you treat with some of these therapeutic agents? And so this is an example of treating one cell line that's quite heterogeneous in terms of differentiation state. On the left, you can see the control. Interestingly enough, when you treat with a MAP kinase inhibitor, what you can see is that the population shifts state almost completely. And so you go into a keratin 14 positive state after treatment for literally for a day with a MEK inhibitor. And if you treat with a PI3 kinase inhibitor, you shift it in the opposite direction. So now you've shifted into more of a keratin 19 or vimentin positive state. And so one of the things that's really important, about that is that when you do that, the pathway usage follows the differentiation state. So it turns out that if you push cells with a MAP kinase inhibitor into the keratin 14 direction, you downregulate the pathway that you're attacking, and you upregulate the compensatory pathway. And so it goes both directions. So as I mentioned in the beginning, one of the things that we have to deal with when we're talking about cancer biology is the fact that the cells that we're trying to manage don't just live in one microenvironment. Especially in disseminated disease, what you have are cells that are disseminated throughout the body, the brain, the bone marrow, the liver, the lungs, etc. And in each of these environments, the cancer cells are receiving different sets of signals. So if you just look at some of these microenvironments, reading the literature and so on, one of the things that you will appreciate is that some of the signals are actually coming from extracellular matrix proteins, insoluble proteins, and so on. Others are coming from cytokines, growth factors, and so on. So both soluble and insoluble proteins seem to be influencing the cancer cells themselves. And so the question is, which ones of these signals are actually important and which ones are not? And how does one actually cut through this incredibly complex environment to identify some of the most important signals? And so the strategy that we've been using to do that, again, is an immunofluorescence-based assay, but it uses a technology that was really pioneered by Mark Labarge called microenvironment microarrays. And the idea here is that one can take these different proteins, soluble and insoluble, from the microenvironment and print them as arrays of proteins, either single proteins or combinations of proteins. And you can literally print, the technology is the same technology that was used decades ago to print cDNA microarrays. The spots are bigger, but by and large you can print, in an area this big, 10,000 or so spots. And the spots and the environment have been engineered so that each spot is big enough to host the growth of maybe 100 cells. And so you can take one of these slides, toss on some cancer cells, either primary cultured cells or established cell lines, let them grow for a while, and then immunofluorescently stain them for aspects of cell biology that you would like to interrogate and read them out using high content fluorescence microscopy. And so just to give you an example of some of the things that we interrogate, morphology and metabolism, cell cycle activity, nuclear activity, chromatin structure, lineage status, etc. So in essence, we can interrogate aspects of biology that are influenced by these microenvironments by selecting the reagents that we use for immunofluorescence staining. Clearly, again, we're rate limited by the specificity of the antibodies that we've got, but there are enough that we are able to get a pretty robust analysis of what's going on. So this turns out to be a complex experiment. I'm not going to walk you through this, but the typical experiments that we're working on these days interrogate the impacts of about 2,500 different microenvironments in every experiment. So we get a pretty broad view of what's happening or what these microenvironments are doing to our cancer cells. And the answer is, I'm not going to go through the data in general, but what you see here are two plots, one on the left showing you sort of a differentiation state plot, and the one on the right showing a S-phase fraction plot. And the data are rank ordered according to impact. And each of these plots shows you results from about 2,500 different microenvironments. And the bottom line, the only bottom line that I wanted you to get out of this particular plot is that the impacts of the microenvironments can actually be amazingly dramatic. So you can literally completely change the differentiation state by changing the microenvironment, and you can move the cells from being fully quiescent to being rapidly proliferative, again, just by changing the microenvironment. So these are all the same cells. They're just receiving different signals from the microenvironment. So that's interesting from a fundamental biology point of view, but we think it also has an impact in the context of therapy. And the reason for that is that not only can we grow the cells on these diverse microenvironments, but we can also treat them with therapeutic agents. And so what I'm showing you here are results that we obtained for one different microenvironment here. These are results for a breast cancer cell line that was HER2 positive, and it was grown in increasing concentrations of lapatinib. Now, lapatinib is a HER2-targeted therapeutic agent, and if you look at the left half of the left panel here, what you'll see is the dose-response curve that we expected. As you increase the concentration of lapatinib, the number of cells that remain in the culture decrease. But if you happen to put those cells in a microenvironment that are rich for neuregulin, one of the things that you immediately see is that lapatinib is no longer an inhibitory drug, it's a stimulatory drug. And so the cells actually grow faster in the presence of lapatinib than they do in the absence of it. Now, that's one cell line on the left where that happened. There's another HER2-positive cell line on the right where it doesn't happen. And the difference between the two is that the HER2-positive line on the left is actually in a luminal-like background, an ER-positive-like background, and the one on the right is an ER-negative-like background. And one of the things that we're particularly interested in these days, again, that will depend on the on antibody availability, is the fact that if you don't use small molecules, if instead you use siRNA therapeutics, one of the things that we discovered is that knocking down the gene actually is robust against this kind of microenvironment-mediated resistance or long-term development of resistance. And so what we've learned is that even though cells may become resistant to lapatinib and trastuzumab and so on by extended growth in these environments, they are not resistant to siRNAs. And so there's been a lot of work in our lab in collaboration with Wasana Yanitsi and around the world in developing now robust platforms that allow us to deliver siRNA therapeutics in vivo. The one that we happen to be using is a mesoporous silica nanoparticle carrier. Again, what we're doing is taking advantage of the fact that these can be targeted to the tumors that we would like to treat using antibodies. And so we've made a HER2-targeted anti-HER2 siRNA therapeutic. And one of the things that you will appreciate is that these reagents are effective in vivo, given by tail vein injection, and they are effective in tumors that are otherwise resistant to HER2-targeted therapeutics. And so it's given us now the ability to think broadly about how it is that we would begin to counter some of these microenvironmental effects, because it gives us the opportunity to pick and choose which are the genes that we're going after. Obviously, still much work to be done to demonstrate clinical utility of this, but at least from an animal point of view, it is exciting to us. Well, this takes us in the direction then of wanting to actually understand what the composition of these diverse receptors are on the surfaces of cells. And I mentioned that there are a lot of new analytical technologies that are coming on board. Nobel Prize in Chemistry was given last year, I guess, for super resolution microscopy. One of my colleagues at OHSU, Xiaolin Nan, is now well along in developing the next generation of super resolution microscope that allows you to measure both the position of a protein and its spectral characteristic simultaneously. And so what this allows us to do now is to image samples that are, with about 20 nanometer resolution at 20 separate colors. And if we had antibodies for 20 separate proteins that were small enough not to have steric interference, we could actually then begin to actually quantify the distribution of these receptors on the surfaces of cells. This raises the question, where's the HER2? Where are the receptors? And so one of the things that we've come to appreciate is that when you use super resolution microscopy, a good fraction of the receptors that are in these cells are actually present on these long protrusions. and we see them in vivo as well as in vitro. So we become fundamentally interested in what they are. They do appear to be not only HER2 containing on the outside, but actin containing on the inside. Here you can see a SEM view of the interactions or the extensions between these protrusions between cells. This is immuno-EM, and so if you're sitting in the front row, you'll see that there are a bunch of white speckles on that protrusion. Those are immunogold labeled HER2 molecules, so they extend between the cells. I'm going to try to skip that and get to the question of, are they doing anything? And so one of the things that we became aware of a few years ago was this work from the Lidikis and Chauvins demonstrating, at least for EGFR, that not only did they seem to be present on these protrusions, but they also seemed to be moving along the protrusions. In their case, they postulated that this was, excuse me, retrograde transport through actin depolymerization. But we wanted to ask the question for HER2, do we see this kind of transport? And so again, we used an antibody reagent to ask this question, in this case a growth factor conjugated, or a quantum dot conjugated to an antibody against HER2. So this sort of brings me to the end of the story. I think that we really are in a situation now where we have a broad range of uses in cancer for antibody-targeted reagents, reagents to deliver siRNA nanoparticles, reagents to quantify the heterogeneity that needs to be controlled as we steer, or as we treat cells and steer them into environments in which they can be more effectively controlled. I think that we do now know that selected microenvironments can dramatically influence therapeutic response, and I think that there's a real opportunity there to develop inhibitory antibodies, not to counter what's going on in the cancer cells so much as to counter what's going on in blocking signals from the microenvironment. And so I think that the other thing that I will point out is that this whole field of multiscale, multicolor imaging is really just coming to the fore at this point. We're critically limited by the availability of validated antibodies, but I think it's a real important need for the field, and hopefully this group will take on that challenge. Thank you.
Yeah, I had a question about your microenvironment studies. I assume those were all human proteins you used?
Yes, those were all human proteins. In essence, we just buy them.
Yeah. So have you ever tried the equivalent mouse orthologs side by side to see how significant just using the orthologs would be?
No, we haven't. Interesting question. It's a little beyond our technical capability. I mean, our technical throughput right now, but we will get there.